First, I would like to thank the organizer, Tsinghua University American Society of Microbiology, especially Dr. Ding Qiang, to invite me. So, and also thanks Stacy for a wonderful background of influenza, so I can skip some of them. But I just want to remind you, influenza is always there, you care or you don't care. So every year, the seasonal influenza caused a lot of infections and severe cases and deaths. And the more important thing for influenza, because influenza virus can cause pandemic. I mean, we have at least 4 pandemics during the last 100 years. So what's the reason for that? The influenza virus is very complicated. So the ecological distribution of influenza virus it's not only globally, it's kind of on the earth everywhere. So you can see the virus can infect different species, and sometimes the virus can transmit from one species to another species. So for my studies, focus on the avian influenza virus. So you can see the list. The avian influenza virus can infect the human. The list is long and they become longer because a lot of new subtypes avian influenza can cause human infection. And also you may see that in this year, United States, the virus transmitted from birds to cattle and cattle to human. But this is it's not a new story. Actually, this virus caused human infection for a long time. As just Stacy showed that the virus host interaction not only caused the affect the interspecies transmission, as also caused the clinical outcomes from asymptomatic to severe in vitro death. And then the virus protein and also host factors interact together. So you can see the list here for the virus, a lot of mutations affected this phenomenon, and also a lot of host factors. So the genes are marked with red is some research studies by our group. So today I will focus on the H7N9. So H7N9 occurred 10 years ago, and actually first detected in Yangtze River region. And this is a triple reassortant virus derived genes from different different subtype of the virus, and caused the 5 outbreaks in China, including 1,500 cases, with the case fatality rate is about 14%. It's a huge, it's a big one. Due to China used the mass vaccination in poultry, and this virus, we did not detect the human cases after 2019. But still, WHO and in this field, they still put H7N9 on the high list for the virus could cause pandemic potential. And also, you can see this figure is the emergence and the impact for H7N9. So you can see, compared with the other subtype, avian influenza, H7N9 is still very, very important. So for this virus, because this virus was first identified by our group, so we have a good surveillance system, so we can see this virus from the wild birds to poultry and then cause a human infection. So we call this procedure called genetic tuning. That means the virus can adapt to the new host and jump from one species to other species. So a lot of factors affect this procedure, but some of the key factor. So for example, for virus infection, the receptor binding is very, very important. So for this virus, you can see the H7N9 virus can bind both receptors. We call the human receptor and avian receptor, and the distribution of two receptors different in the respiratory tract. And this phenomenon is only caused by 2 mutations of HA. So that means this is very important. And also for the clinical outcome, 2 mutation for BB2 also is very, very important, because if these 2 mutations happened and the virus became more virulent and it kills mice in animal model, and also increase the transmission of these 2 mutations happened. So the first question is if this, how this virus infected the human due to the receptor binding. Another one is other host factors play a role for this. So when we look at the epidemiology data, we see that the liver poultry market is the main infection source, but we compared the normal population or regular population with the poultry market work populations, we did not see difference. That means even people exposed more, but they didn't have higher risk for that. And so we think some important host factors play a role. So we did a cohort study, and including the cases and the control with the same exposure history, and to do the genomic study. So because we are familiar with this, I will skip that. So we use the different analysis method and try to find the host factors. So this is the cohort summarization of the cohort. So we include the H7N9 cases and poultry weight controls. And after that, we use 2 general population for the analysis. So if we just using the GWAS association analysis, we didn't find any positive signal. But when we use the gene-based association, we found the MX1 gene played a very important role. So you can see the OR is very higher compared with the general population. So that's exciting because MX1 recognized by long time is the antiviral genes play a very important role against not only RNA virus, also DNA virus. And also the structure also solved by research groups. So this virus, this protein can divide into different structure, including the G domain, the BSE region, and the stalk region, and the loop. So when we put back all the mutations we found, we didn't see a specific distribution of all these mutations distributed around this protein. But indeed, for some regions, so for example, BSE regions, they showed a higher risk. So we think that region may be more important. So then we tested these mutations functions using the in vitro. So firstly, we tested the polymerase activity using the mini replication system. So you compared with the wild type, you can see if the mutation happened, it lost the antiviral function. And the same thing happened if you test the virus title. So confirm that if mutation happened, this the antiviral function lost. And then we tested different viruses, not only H7N9, we also test the H1N1 and the H5N1, H7N7, the similar results obtained. So that means this MX1 gene is true important for the antiviral functions. And then we look back in the human samples. When we look back all this MX1, the mutation in all patients we detect is heterozygous. That means these patients containing a normal copy of the gene and a mutation copy of the gene. So if what's the exact in the human? So we test these 2 copies. One is containing the mutation, and the other is the regular. So we put them together, co-transfected, and we can see here if we when we add more mutation copy genes, so you can see the viral inhibition function lost. So that means it's a dominant negative for these mutations. And also, if you test the virus title is the same. And also, we confirm these two proteins can interact together. So another story I want to share is the sex play a role, because when we look at the epidemiology data, we found that the male is the majority of these patients. Is about 17%. Of course, a lot of factors can contribute to this phenomenon, because, for example, the professional poultry exposure is essential. Then we look back: is this true for the male? Was the dominant the majority? So we use the same cohort and to look at the sex hormones level and the cytokine response for male and for male and female, and the results showed that they reduced the testosterone level is associated with the infection and the severity of the ASM in man, but not associated with the estradiol levels in man. And then we collected another cohort infected by seasonal influenza. Actually, the similar results obtained, and the reason maybe is due to the inflammatory cytokine and the chemical response associated with this phenomenon. So we tested a lot of cytokines and chemicals, and not only for H7N9, but also collected the data from seasonal influenza, and we found that some markers, such as HL15, are negatively associated with the testosterone levels in H7N9 in infected man. So that means some reason caused this due to the this one, also we tested in women population, but not. And then we wanted to replicate this kind of phenomenon in the mice, but this is not an easy model to do. We tried a lot, but finally we found that H7N9 can replicate in the natural cells and reduce the circulating testosterone level in mice, but this is not the case in human, I believe. So, but at least indicating that during the virus infection, I mean, something associated with the hormone levels, and they enter together and they cause the clinical outcomes for human information. But at least the cytokine response very similar with the human patients, as we see the previous. But you can see this infection not cause severe damage for in the test. So that means only to say that means during in the human patients is more complicated than the mouse study. But it's so we do not have a good model to study the mechanism. But if we wanted to understand the exact mechanism, we need to develop a good model for that. So that's from the host factors. Some host factors played a very, very important role for that. And recently, based on the cohort I just described at the beginning, we found that the gut microbiota also played a very, very important role. Even this is a very preliminary data, so we use the cohort information. You can see some play the protection and some in the reverse side. So that means the virus host interaction not only involve the host factor and also the virus host, but also involve the environmental, we call it environmental of this during this procedure. So in summary, from this bacterial study, we found at least 12, and some showed the increased risk, and some showed the protection, reduced risk of H7N susceptibility. So the mechanism is very complicated. We don't know yet, but we found some clues. So some network identified, so as shown here. So maybe some network interaction network played a very, very important role for that. So this is a very preliminary story. I'm not a bacteria, so I don't know nothing about the bacteria. So I really hope I can work with other groups. Yeah. So in summary, so as I showed here, we found 12 MX1 variants, and it saw dominant negative effect on the antiviral function of MX1, which increases human susceptibility to zoonotic H7N9 influenza virus. And also the hormone level effect may be associated with the clinical outcome during H7N9 infection and also seasonal influenza infection. So, and the gut microbiota were casually associated with H7N9 susceptibility, and we did more study for that. So finally, I will stop here, and I wanted to thank the colleagues and the collaborators. So MX story collaborated with Professor Martin Schumer, and the Holm story collaborated with Gushi Gabriel, and thanks the funding and for any questions. Thank you very much.
So we have time for one or two questions. Is there any question? Beautiful work, as always. Your data is really interesting about the low testosterone levels, especially in that age range of men. Do they have any other comorbidities? And I ask because we see with that increasing body mass, you see a significant decrease in testosterone levels.
Yeah, so that's a good question. Actually, yeah, but we don't have an answer for that, honestly, because for H7N9 cohort, we don't have a case anymore, so we cannot follow that. We try to find, using the influenza, seasonal influenza cohort, but based on the preliminary data shows that for the seasonal influenza, not that significant. So maybe I don't know. Yeah. So yeah. That's it. Yeah.
Okay. And very, very interesting work. I'm really surprised about this very specific effect of MX mutations on H7N9. So because H5N1s, they use this avian receptor. So why would this be specific to H7N9 since they use both receptors? Do you have any hypothesis why it affects more this infection by H7N9 compared to other zoonotic strains of influenza?
You mean the MX1?
Yes.
Yeah.
Yes.
We believe MX1 play a role not only for H7N9, because we have the data showed that it's also affected the antiviral function when we use the H5N1.
Okay.
And also use the seasonal influenza. So we think it's a regular.
Okay.
Yeah, something. Yeah.
Thank you.
Okay, thanks, Dr. Shu.
Okay, thank you.
Yeah, thanks.